Okay. So yeah, I started recording the class lectures before we did chapter 4. So I figured I would just by myself, all alone, just record me going through chapter 4, get through as much as I can get through before I got to stop to pack up for the day. And yeah, so I'll hop into it. Going over a tour of the cells. Let me get my cursor over here. And so here's the idea. All organisms are made of cells. The cell is the fundamental or the basic unit of life, is what you also hear called. Meaning that, you know, back in the day, biologists would take like a living organism. They would see, okay, that living organism is made of cells. They would then take a sample for some other living organism. See, it's made of cells. And they keep doing that enough until eventually you can kind of conclude that everything is made of cells. Technically, we haven't looked at literally every single organism under a microscope, at least I don't think, and seen it has cells. But as far as we know, every living thing we made of cells. The cell is the simplest collection of matter that can be alive, meaning you can take a cell from like your cheek, put it into a plate, and that cell will be able to survive by itself, assuming you give it nutrients and stuff like that. Whereas you can't like remove the Golgi apparatus from a cell and let it be self-sustaining. And all cells are related by the descent from earlier cells, meaning all cells come from a pre-existing cell. You know, and then the idea would be that there'd be some ancestral cell that all cells came from. And yeah, all cells are going to have some common features that we'll go over in chapter 4. And even though like you could look at like a nerve cell compared to like a red blood cell, they look completely different, but they still are going to have those same features, those same things in common. Okay. So 4.1, biologists using microscopes to study cells. I'm going to go over this pretty quickly because I'm not really going to ask you any questions from 4.1. It's just kind of going over briefly just some of the different kinds of microscopes. So that's something, if you're really curious about, I'll let you kind of read that further in your book. But yeah, basically cells are really too small for us to see with the unaided eye. Unless you're talking about like some elephant nerve cell that's gigantic, you have to have a microscope to look and be able to actually see these cells. And yeah, the basic microscope that we use like in school and all that is the light microscope. And the idea is you're going to pass light through your specimen and then focus that light using lenses, and then you'll get some sort of magnified image. And those lenses, they bend that light, and the bending of the light is how we'll then be able to magnify that image. When we're talking about microscopy, there's 3 different kind of categories or parameters we use. Look at magnification. So basically just sort of how big is the object, how zoomed in. Resolution is the clarity, so the distance between 2 points. So you can think of resolution like if you were like, you know, you keep doing this and get your fingers closer and closer together. At some point, or if like another person was doing it with their fingers, there would come a point where you can no longer tell a difference between the 2 points. And that would be, even though there is clearly two different points, the resolution is too small. So it's a similar thing with like photos. You know, what makes one photo like really blurry, like, you know, older photos versus, you know, with the new iPhone, is that resolution ability. And then contrast is the difference between light and dark areas of an image. So sometimes images don't show up very well because there's not a good enough contrast. Like everything is too bright, so nothing stands out. So we'll use dyes to help us get a contrast, because cells, if you don't dye a cell, it's really just transparent. Light's just going to go right through it. So we'll use dyes and stains that will then give light something to bounce off of and refract off of, and then that can be magnified and give you an image. Light microscopes, I mean, they're great. They're pretty cheap, relatively speaking, but they only get us to about 1,000 times the size of the image. And so I'll show you the electron microscopes you'll have to use to get closer. And the staining techniques I talked about, that's what this is saying. And then, yeah, so really to get beyond and start looking at the organelles, that's where you're really going to start needing like the electron microscopes to see them closer. I love this slide. I'm not so much going to like, what is the meter diameter of a human egg? I'm not going to ask you that, but I think to really get a good understanding of biology, it's a really important slide to kind of get your mind around. And then, you know, just like the idea that atoms are super, super small, 0.1 nanometers, all the way up to human height, and just kind of getting a relative feel for how big things that make up the cell are. And then, you know, this is kind of this idea of like, what can you see under a microscope versus what do you need an electron microscope to see? Okay. All right, so getting to the electron microscopes, as I said, I'm not going to make a huge deal out of it. I think understanding these two basic definitions, that's useful for just your general biology knowledge, where if I'm just the scanning electron microscope, basically you'll like coat your specimen with, I think a thin sheet of gold is what they use, some sort of metal that can reflect, you'll shine a beam of electrons at your sample, and those beam of electrons bounce back from your specimen. And then based on like kind of the angles that they bounce off the specimen, a computer can like construct a 3D image of whatever it is you're looking at. Whereas a transmission electron microscope is sort of similar to a traditional light microscope, except instead of a beam of light, it's a beam of electrons. And before I move on, the reason why like a beam of electrons is better than a beam of light is the wavelength that's being utilized. So like the wavelength of light has too big of a wavelength for you to get too much, like there's a limit to the resolution you can get, because light, the visible light, only has a certain nanometer, whereas a beam of electrons has a much smaller wavelength, meaning you can get a much smaller resolution. You can get down to a smaller level. So yeah, if you're going to look at the internal structures of cells, transmission electron microscope is really helpful for that. So 3D image scanning electron microscope, internal structure of cells to transition electron microscope. That's really about all I'm really wanting you to get from the different kinds of microscopes. You know, you could read this in the book. I'm not going to make a big deal about it. Just kind of understand these are just same cell, just different ways you could show it using, depending on what kind of microscope you're using. Fluorescence is pretty important. So within light microscopes, we can tag molecules with fluorescent dyes. So say you're going to build say, okay, you have like DNA, you could tag DNA with these radioactive molecules, or tag the cytoskeleton, here they did in green. You could tag that with this fluorescent molecule that when you shine light on it, it'll then give off this glow. So there you go. Some cool pictures of cilia with scanning electron versus transmission electron micrograph. Don't need to make a big deal about it, just kind of, you know, FYI, these are things that exist. Yeah, so I mentioned this. It's kind of interesting, cryo-electron microscopy. So it's still an electron microscope, but if we preserve the specimens at a really low temperature, you can get a more realistic structure what the cell looks like. Because what happens usually whenever you prepare, like you can't just take cells and just like throw like immediately off an organism and put them on a microscope. Usually like the staining process that we use to get the cells, or if it's a light microscope, the staining process, or if it's an electron microscope, the traditional techniques you have to use to get that specimen ready to be used in the microscope will kill the specimen, and it can alter kind of subtly the actual structure of what's going on in that cell. So this is where cryo-electron microscopy is useful, because by freezing at an extremely low temperature, it helps better preserve the actual true structures of things. Meaning in normal electron microscopes, micrographs, you might get a picture and maybe you think, oh my gosh, that's like a new organelle, or that's something cool or whatever, but you don't maybe know if like, oh, that's actually an organelle or some key cell part, or if that's just like an artifact or something in the image, something that wasn't supposed to be there. So anyways, that's that. Cell fractionation, this is a pretty useful idea. And this is really helpful in just all kinds of lab work where they're trying to understand like what chemical reactions are maybe happening in different organelles. The idea is you'll take your sample and you'll put it in a test tube, and then you put it in this machine called a centrifuge. And this centrifuge is going to speed that test tube super, super fast in a circle, around. And the idea is it basically will separate out that cell based on different weights. And why that's helpful is within each density level, maybe like say like I don't think of a picture of it, but I can draw something real quick. So say this is your test tube. Maybe like in this layer here, like the very bottom layer, maybe that represents, let's say, the mitochondria or something. And then, you know, you have this layer here, this represents the Golgi apparatus, hypothetically. So you can separate out, and then you can separate out the mitochondria from everything else, and then be able to have like this layer mitochondria. And then you can analyze, okay, within this layer mitochondria, oh, we found these enzymes, and these enzymes are really important for cellular respiration, hypothetically, right? So that's the idea. It's kind of a useful lab technique. We don't get to do it here just because we don't have the centrifuge machines or enough of them that work well. But yeah, important idea. All right, 4.2 is really the meat of chapter 4. And, well, I would say the meat. 4.2 is really where I'm going to start testing you on chapter 4. 4.1 was just kind of there, to be honest with you. But this is getting into prokaryotic cells versus eukaryotic cells. So basically, eukaryotic cells, they say they have these internal membranes and compartments. So that's talking about the organelles. So eukaryotic cells, this is how I always think of it. Eukaryotic. You pronounce this EU as, this is pronounced as a U, and this is carry, but like they aren't very good at spelling, I guess. So you carry a nucleus, or you carry the membrane-bound organelles. I'm talking about the membrane-bound organelles. So you carry those, and then I feel like you need to get like fake crowd noise or something, like a laugh track. And then pro means no. So prokaryotic, or pro rhymes with no, I guess. So pro, think no, as in they have no nucleus and no membrane-bound organelles. So I think it's the simplest way to remember that. Yeah, and every single cell is going to be made up of either a prokaryotic cell or eukaryotic cell. So one of these two categories. Within prokaryotic cells, there's either bacteria or archaea. Bacteria, I feel like we're fairly familiar with those just from everyday life. But archaea, these chaea, think chaos. So these are bacteria that live in like chaotic environments, like super salty or extreme heat. So yeah, and then eukaryotic cells, fungi, animals, plants, really about everything else. Protists, think of protists are basically like bacteria that are made of, they're single celled. They're kind of like bacteria, but it's like a bacteria made of a eukaryotic cell. It's a bacteria that would have a nucleus and membrane-bound organelles. It's sort of the simplest way. Protists is the most squishy category, where it's like the category where like we don't really know what this is. Let's just call it protist, I guess. All right, moving on. So you gotta know all the features that all cells have. And I think these are somewhat common sense, like the more you think about it. All cells have a plasma membrane or a cell membrane, meaning all cells need to have some way to separate the inside from the outside, right? Some way to separate inside from outside. And all cells are going to have some sort of fluid that the parts of the cell can float around it. That's going to be your cytosol, or also called the cytoplasm. So that makes sense, right? If you don't, it's easy to take the cytoplasm for granted, but it's super important, because if you don't have a cytoplasm, or if you have too much cytoplasm or not enough cytoplasm, that's going to change the rate of those chemical reactions. And if those chemical reactions go too fast or too slow, that's going to affect the health of the cell. And then all cells have to have some way of passing on their genetic information or carrying that genetic information. And so all cells are going to have some sort of chromosome. Now, if you're a prokaryotic cell, you have a circular chromosome. Prokaryotic, they have a circular chromosome. I don't know, like circular. Circles look like this, and there's a lot of more circle-looking things in the word prokaryotic than eukaryotic. I don't know. I don't know what you want from me. But like DNA is the double helix for us. So eukaryotic, that's when you're the double helix. In other words, they're not all DNA is double helix. You carry a nucleus and membrane-bound organelles, and if you're really cool, you also carry heles, like the tennis shoes with the wheels in them. That's how you can remember that. I don't know if that's how you spell heles, but there you go. Okay, proteins. Proteins are like the workers of the cell. They're super important to the functioning of the cell. Most of the mass, like the dry mass removing the water of a cell, is proteins. So that means you got to have, all cells have to have some way to make those proteins. So that's why all cells are going to have ribosomes. Okay. All right. Moving on. I think I've kind of said everything here, all right, with the pro and no and eukaryote and all that. Yeah, a little additional detail. The DNA, the prokaryotic cell, it isn't just floating around willy-nilly. It is technically in this region called the nucleoid here. So yeah, nucleoid is kind of like this protein bound region in the prokaryotic cells, whereas the eukaryotic cells, they actually have like a double membrane bound nucleus, which I'll show you a little bit later in chapter 4. Okay, so here's your prokaryotic cell. You know, if I showed you this cell, you need to be able to say that's a prokaryotic cell, not a eukaryotic cell. And then you need to know these different parts of the bacteria. So these little hair-like looking things here is called the fimbriae. So these are going to help attach their attachment structure. So if a prokaryotic cell, when it wants to attach itself to some sort of surface or onto another prokaryotic cell or a eukaryotic cell, the fimbriae are going to help it attach itself. And then going more inward from that, you're going to have what's called the Glyco Calyx. So the Glyco Calyx is that very outer coating. So it's what's in this yellow here. And it's also called a slime layer. So they call it a slime layer because it's slimy, I guess. Then you have flagella, these little tails. Locomotion, it's just kind of like a fancy word for motion. It's helping move around. And then, so going more inward from the glycocalyx, you then get to the cell wall, which, you know, plant cells, fungi, they have cell walls. You know, obviously animal cells don't. But then you go one more layer in, then you have the plasma membrane. So you're familiar with those. Ribosomes you're familiar with. Don't forget that that's a very common thing. People tend to think that a ribosome, some places they'll call a ribosome an organelle. The reason why that doesn't invalidate the pro and no is because membrane-bound organelles. Ribosomes, they're organelles, but they're not membrane-bound. There's not a cell, there's not a lipid, like a phospholipid bilayer that surrounds the ribosome. Okay. And then here's the nucleoid, that region where the DNA is at. Notice how it's circular. Well, I don't know. It doesn't really look circular there, but like if you were to just kind of take my word for it, if you were to like take it out, it'd be a circle. Okay. Eukaryotic cells are a lot larger than a prokaryotic cell. So like looking at these ranges, even if you would take the biggest bacteria and the smallest eukaryotic cells are still twice as big as a bacteria. Or a eukaryotic cell could be up to 100 times bigger than a bacteria. So let's say 20 to 30 times bigger than a bacterial cell. It is the diameter of a eukaryotic cell. And what's interesting is, you know, we tend to think of bacteria as these bad things, like, oh, you know, I'm sick from this bacteria, E. coli or whatever. But you actually have more bacteria that live on or inside you than you have actual eukaryotic cells. And that's a huge area of research, is trying to understand how do like the good bacteria, we call them, we know that they interact with our different organ systems to help keep us healthy. And just trying to figure out how exactly they do that, or what species of bacteria even we want to promote in different parts of our body. For example, they've analyzed like the stool samples of somebody with like chronic depression, and the stool samples of people who don't have chronic depression, and they'll see that there are some like certain like strains of bacteria that people with like certain mental health issues are clearly missing. So and I'm not really too sure if there's like a chicken and the egg kind of thing going on with that. but clearly there's something going on where like the bacteria in your gut can help. They call it like the gut-brain axis. They definitely know that the bacteria in your gut can help in some ways in keeping our mental health. So there you go. All right, plasma membrane. We're going to talk more about that in chapter 5, or as you're watching this video, you already have. It's a selective barrier. I mean, it doesn't let anything, it wants to be choosy about what it lets in and what it lets out, or when it lets things in and when it lets things out. So usually waste is a thing that's going to be leaving. Yeah, and remember, it's a phospholipid bilayer, so 2 layers. So here's your cell membrane. So they have a hydrophilic and a hydrophobic region. So notice how the gray heads of the phospholipid are part of the hydrophilic region. If I shade that in, they're part of the hydrophilic region. On the outside of the cell, this is outside of the cell, and this is inside the cell. So the hydrophilic heads, we would also call these hydrophilic. We also refer to that as polar, and hydrophobic non-polar. So these heads here, these are polar heads. So if you remember that structure of the phospholipid, you had that phosphate group and the choline group that was polar. That's why it can interact with the water on the outside and the inside of the cell. And then the hydrophobic region, this is why you have those fatty acid tails that were that long string of like carbon to hydrogen. You know, if I were to draw it out. So anyways, hopefully that's kind of second nature to you by now. These carbohydrate side chains, these are really important in cell recognition that you'll learn more about in chapter 5. But they're almost like the fingerprint of cells. So, you know, how like, say blood typing, you know, some use like blood type A, blood type B, they'll have different carbohydrate side chains on the outside of their cell membrane, and that will, you know, give the presence of what we call type A or type B or something. And so that can be a key thing our immune system can use to recognize like foreign invaders, or even like within a tissue layer, how like cells within a tissue layer can recognize each other and part of how they can work together with one another. Then throughout the cell membrane, you have these proteins, and these proteins do a bunch of different jobs. In chapter 5, I'll go over some of those jobs. But yeah, there you go. Keep this thing moving. Okay, surface area to volume, really important concept. Surface area, that is the area of the surface, turns out. So surface area is like, you know, if I were to color this cube, and if there was an actual cube that I was holding, you know, this isn't a cube, but it's a remote that is a rectangular prism, I think. The surface area would be like, if I were to paint this, the amount of paint I would use to cover the whole thing. Whereas the volume is the space on the inside. So something interesting happens. Surface area, let me give you some space to write. Surface area will not increase as fast as volume. So let me write that down. So surface area increases slower than volume. So here's the consequence of that. You know, when you went through puberty, right? You're growing, most of you, and you got bigger. You may think, well, did I get bigger by all of my cells just getting bigger? And that might seem to make some sense, but that's not really what happened. You got bigger not because your each individual cell got bigger. You got bigger because you made more cells. And the reason for that is because the surface area increase is slower than the volume. Meaning, if you look at this, you know, this giant cube here, the volume here is the same as if I took this same volume and I just broke it up into a bunch of smaller cubes. The difference is I would have more surface area. I could then, by breaking up into smaller cubes, you have way more surface in between the actual interior. And then the purpose of all that, having a smaller, we have a surface area to volume ratio. The idea is if I have a cell with too big of a volume and not enough surface area, it's like a school not having enough doors or something, right? You increase the volume of the school, but you forgot to add more doors. you're going to have a difficult time getting people in or out during like a fire drill, or think of the hallways, you know, at McLean, you know. Whereas if you make the cell smaller, you're going to have, you know, you have the volume to the surface areas is appropriate. You have the right amount of doors and hallway space for the volume of the school. Okay. And related to this idea is folding. So the reason why when you look inside of a cell, like if you look inside this cell, you see all these folds. Like here's the smooth endoplasmic reticulum, it's got these folds. Rough endoplasm, it almost looks like a maze. All these folds, or even if you look at a full organ, like the small intestine, it has all these folded like structures. And then even within one small intestine cell, if I draw it bigger, it has all these little microvilli extensions. And the idea here is all of these extensions and folds and stuff, that increases your surface area. That gives you more space to bring things into the cell or to release waste. That's why surface area is important. We want to be able to bring things in and let things go in an efficient manner. Okay. And if you have too big of a volume for your surface area, you're creating too much waste, or you need too much nutrients, more weight, you make more waste and you need more nutrients than the cell membrane can handle with like processing. Okay. This is also why if you look at, this is the very end. You know, so if you look at your lungs will keep like branching almost like a tree until you get all the way down to the very ending point where you get to what's called the alveolus, which is this like, looks like a cluster of grapes, this very sac-like structure. But between all of these sac-like structures, you have the reason why it's not just the reason, in other words, the reason why it's not one big ball and it's multiple smaller balls is that's going to give you more surface area while maintaining the same volume. Okay. So having more surface area, you then be able to have more blood vessels that can wrap itself around that alveolus and exchange the oxygen and carbon dioxide that we need to keep ourselves healthy. Yeah, so the metabolic requirements, meaning like the amount of nutrients you need to bring in and the waste you need to get rid of, that's going to set the limit on how big a cell can be. Yeah. Okay, set everything there. Here's another example if this helps, just kind of, you know, here's like, okay, if you had 1 cube, the surface area, let's say it's 6, volume is 1, surface area to volume ratio. So you do surface divided by volume, 6 divided by 1 is 6. Let's say I have 1 giant cube. Okay, surface area is 150. Yeah, so this is you like, oh, I just got bigger by making all my cells bigger. Okay, if you try doing that, you would have a surface area of 150, volume of 125. 150 divided by 125 is only 1.2. So this is a problem because you're not going to have enough cell membrane space to bring in the nutrients your cell needs, or enough cell membrane space to get rid of the waste the cell's making. But okay, if you still need to get bigger, right? You need to be all big and strong and stuff. So you got to find some way around that. Well, keep the same volume. Like if you need to be 6 foot, right? Your genetics demand that of you. Well, you can be 6 foot, but just make all of your cells be smaller, right? So just take that volume and just break it up into smaller cells so that you can keep that surface area to volume ratio constant. On the AP exam, they give you these. This is just like a Snagit picture from the AP formula sheet. So they'll give you volume of a sphere, cube, and all that. You just need to be comfortable doing some surface area to volume calculations. If they gave you a cell and said it approximated a column, they probably give you a sphere. You would need to use the radius they give you, find the surface area, then find the volume of that sphere using the radius, and then calculate the surface area to volume ratio by dividing the surface area by the volume. Okay, so getting into some of the organelles. Basically, what they mean by cells metabolism is in order to create energy that your body needs, or make those different biomolecules you learned in Unit 1. The cell has to have the plasma membrane and the organelles working together in harmony. The cell is like this beautiful factory with all these different organelles working together. So for 5.1, this class, I didn't at some point when you take the test, you need to know the function of all of these different organelles. It's a good review slide. But in class, I didn't actually go over these in detail. I just kind of showed it to you. It was just a picture that your book had included for 4.1. Okay. Notice all the folds that they have. You need to know the difference between the animal and the plant cell. So notice how the plant cell is going to have this one giant central vacuole, whereas animals, they don't have one giant central vacuole. They're going to have multiple smaller vacuoles. So I don't even know if they show a vacuole in the animal cell. Yeah, I don't even see it. Whereas, yeah, so pretend that there's some small animal. And the reason why plants are going to have one giant central vacuole is plants, plants just like stand around doing photosynthesis like idiots, right? They can't do this or like go to the grocery store or the water fountain. So a plant, it needs to, if it's going to rain, it's going to get water. It needs to really be able to store all of those nutrients because it doesn't know when it's going to get its next supply of water. Whereas animals, we don't really need to waste as much because, volume is money, I guess, in the cell, right? Like it doesn't want to waste space. Animals doesn't need to waste space on a huge central vacuole if that's going to, prevent it from maybe having more space for mitochondria to make more energy or something to like hunt bears, I guess. Another thing is you'll notice that there are no lysosomes inside this diagram. So only the animal cells have lysosomes. The lysosome is like the trash can of the cell. I'll show you it in a little more detail in chapter 4, along with these other parts. Okay, so that's 4.2. I'll keep moving on to 4.3. Yeah, so this is getting into the details of the nucleus. So remember, you carry a nucleus. So inside the nucleus where we carry the DNA. And we also will make the ribosomes in the DNA. We also make the ribosomes in the nucleus. We make the ribosomes in a part of the nucleus called the nucleolus. It's just kind of sort of a center-ish part of the nucleus where we're going to make the ribosomes. And remember, the DNA, it has the information for the proteins. So on the DNA, you have genes, and every gene has the information to make one protein. And the ribosomes are what make the protein, just to be clear. Okay, so I talked about that. It's the most conspicuous organelle, meaning the classic way you can tell a prokaryotic cell from a eukaryotic cell is you look for that nucleus. You see a nucleus, that's a good indication that cell is going to be a eukaryotic cell. Yeah, the nuclear envelope and the nuclear membrane. It's a let me get to a picture of it. It's not just a lipid bilayer, it's a double lipid bilayer, meaning I don't know if you can see this super well. I can just draw it briefly here. You'll have those phospholipid heads. Here's your hydrophobic tails, and then you have like the heads, and that's your normal cell membrane that I just drew here. But like what's unique about the nuclear membrane is you then will have like this inner membrane space, and then you do the whole story over again. You then have another layer, another phospholipid bilayer. Geez, use your imagination at this point, I guess. But that's what's going on with that. Yeah, a lot of good stuff going on with this picture. Nuclear envelope is a double membrane, but it also has these little pores. So that's what these are. So it's a little hole in the nuclear membrane that will allow, whenever we make the mRNA, that'll allow the mRNA to go through those nuclear pores. And then that messenger RNA will go into the cytoplasm where it meets up with the ribosome, and then the ribosome will make the proteins. So you have nuclear pores. It's super complicated. It's a really impressive filter. Because think about it. You need to let some things come in and let other things go out. You don't want everything to go. So but it has to be efficient at doing that, right? Otherwise, you're just going to get this long security check-in line. But it's super efficient at filtering out what is either too big or not, doesn't belong in the nucleus versus what belongs in and needs to come out. These are not as important, but like these histone proteins. DNA, if you unwind it, it's like 5 feet long. is kind of crazy, something like that, or maybe like a few feet. It's really big, and your cells are really small, and the nucleus is even smaller. To make it fit, all that DNA fit inside the nucleus, you have to wrap it up around these histone proteins, and it kind of reminds me of like a laptop charger, where this is the DNA wrapped around the histones. This should be like the DNA unwound from the histones, where the histone would be the central charger power. I don't even know what this is, the power block part of the charger. Okay, then it's okay, we gotta get it to fit it inside the nucleus. We'll then wrap and unwrap it. And if you ever heard of epigenetics, epigenetics is this new field of study where it kind of investigates like how could like different exposures to our environment change how we wind and unwind our DNA. Because you'll unwind the DNA to make proteins, to access genes you need to make proteins, and you'll wind it back up when you don't need to use those genes to make a protein, so that you can keep it stored inside the nucleus. Anyways, and we call the DNA organized around the histones, call those chromosomes. The complex of DNA and proteins, that's chromatin. You'll kind of hear chromatin and chromosomes used interchangeably. Technically, like the chromosome is like the most condensed form of chromatin. There you go. I mentioned the nucleolus before. Oh, let me point it out. This would be the nucleolus in the diagram. So it almost looks like I may be making this up, but the nucleolus is like the nucleus of the nucleus. I'm going to pretend like that's true. But anyways, that's where we're going to make the ribosomes. Okay, go into a little bit of detail on the ribosomes. Ribosomes are technically made of this thing called ribosomal RNA. You'll hear it called rRNA, where the lowercase r stands for ribosomal RNA. But it's also made of protein. So it's kind of ironic, I guess, or wild that ribosomes make proteins, but they themselves are made of proteins. So you kind of get this whole chicken and the egg thing where it's like you got to have ribosomes to make proteins, but then the proteins, and it's kind of interesting. But anyways, ribosomes can be in two different locations, and you need to understand why they're like, okay, if you're a ribosome that's free in the cytosol, this is going to be crazy. Follow me here. If you're a ribosome in the cytosol, free in the cytoplasm, you make proteins that stay in the cytoplasm. Because efficiency, right? Like if I grow peaches, it wouldn't make sense for me to travel to New Zealand to sell my peaches, right? Like people in New Zealand are probably going to buy peaches from people in New Zealand, assuming they can grow peaches there. But like the point is, the free ribosomes that are just hanging around in the cytoplasm, they're the ones that make proteins that stay in the cytoplasm. Then we'll have we have something called the Endoplasmic Reticulum, where we have these bound ribosomes on something called the rough Endoplasmic Reticulum, and I'll show you that in more detail, or rough ER, in a bit. But the rough ER, those ribosomes, those bound ribosomes, they're going to make proteins that for outside the cell. Just generally speaking, so kind of understanding the general idea of the protein, of the destinations of proteins made on those two different kinds of ribosomes. Okay. And here's a picture of a ribosome, and then showing the free versus bound ribosomes. So free ribosomes are just hanging out in the cytosol, and then this is the endoplasmic reticulum, and it's in blue here.
Good afternoon, McLean. It's time for the afternoon news show. Tune into WMHS News on YouTube. Also, the mathematics interest meeting after school today is in Mr. Persinger's room.
Mr. P. Big math guy, Mr. P. Okay. Anyways, here's your endoplasmic reticulum, and then the ribosomes are just like attached to it, bound to it. And with the ribosome, you have a large subunit, a small subunit. That's important to know. Large subunit, small subunit. As you get into college, that like, and you look at like the structure of like a prokaryotic ribosome versus a eukaryotic ribosome. I mentioned at the very beginning of this video, like that whole cell fractionation idea. Oh my gosh.
One quick announcement for the new show, and come out and support our senior field hockey team tonight. It's senior night for them. Game time will be about 7:30, but the senior festivities start at 7. Come out and support them, and also our volleyball team at home tonight against Langley. Thank you.
You're welcome. Okay, so how they build the difference between like a prokaryotic and eukaryotic ribosome and how they'll name them, like they'll use like, I'm making this up, like the 30S subunit, where the S stands for sedimentation, but basically it's referring to like when you do that cell fractionation and the centrifuge, it's like what layer does that ribosome end up being? What basically how dense is the ribosome of like a prokaryotic cell versus a eukaryotic cell? Anyways, not super important, but I just felt the need to say that. Okay. I think this would be a good spot to leave it off. The bell's about to ring here, and 4.4 is kind of longer, so I'll make a separate video going over some of the other stuff in chapter 4.